Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing CSIR pyqs December 2023 shift 2. This is part C questions that I will be discussing. I will be discussing questions on applied biology, methodology, biochemistry, cell biology, cell signaling, DNA replication, protein synthesis, genetics. If you are new to my channel, please do like, share and subscribe. Coming to the first question, if the length of a single continuous alpha helical polypeptide is 108 angstrom, which of the following statement is true? Okay, the distance between two amino acids in an alpha helix is 1.5 angstrom. So 108 means 108 divided by 1.5, that will give you 72 amino acid, and that gives the answer as option 1. The alpha helix contain 72 amino acids without proline and glycine. If proline and glycine are there, there would be a helix breakers. So proline and glycine are usually not found in the alpha helix. Then next one. Which one of the sequences given below is a portion of a potential microRNA precursor? So microRNA forms imperfect stem loop and it is seen in option 4. If you see option 4, you can see U pairing with A, then A pairing with U, G pairing with C, again G pairing with C, G, U, that is not pairing because it forms imperfect stem loop, so naturally pairing will not be there. Then G pairing with C, U pairing with A, then again it doesn't form a stem properly because it is not proper pairing, it is imperfect pairing, and this is the loop. So microRNA precursor is option A, option 4. So if you see option 3, you can see G pairing with G, which is not possible, U pairing with A, okay, U pairing with U, again not possible. So if you see this one, A pairing with C, not possible, U pairing with U, not possible. If you see 1, it is G pairing with U, T pairing with C, again not possible. So the correct answer is option 4. Then next one. Which one of the following options correctly depicts the stereo conformation of the dipeptide derivative of aspartic acid and phenylalanine? So this is aspartic acid, this one. This is aspartic acid, this is phenylalanine, and this is the methyl ester. This one. So now if you want to see, they have given L aspartyl, L phenylalanine. So here aspartyl and phenylalanine methyl esters, they are common in all the options. You have to just see which is L and which is D form. Now here if you see L amino acid, when L amino acid is formed when the NH2 group is on the left side, and D amino acid is formed when the NH2 group is on the right side. So here if you see aspartyl amino acid is on the left side, similarly phenylalanine amino acid is also on the left side. So it is L aspartyl, L phenylalanine methyl esters. Answer is option 1. Then next one. Given below are inhibitors of electron transport, column X, and their target enzyme, column Y. So inhibitors of electron transport in column X, respiratory chain complexes in enzymes in column Y. So dicyclohexyl carbodiamide and oligomycin, they are naturally ATP synthase inhibitors. So A2. A2 is found in only option 2. So option 2 might be your answer. We will see the others. Then B1. Rotenone and demerol. NADH coenzyme Q reductase. Thenoyl trifluoracetone and carboxyl. That is succinate coenzyme Q reductase. And cyanide and azide for cytochrome C oxidase. So the answer here is option 2. Then next one. Newly identified protein X, Y and Z are associated with endoplasmic reticulum membrane fraction. The above endoplasmic reticulum membrane fraction is subjected to high salt treatment. Buffer pH 7.4 and salt is 0.5 molar KCl, followed by fractionation by centrifugation into soluble and insoluble pellet components. The following observations were made from the above experiment. The protein X and Y are fractionated into soluble components. Protein Z fractionated into insoluble pellet component. Based on the above observation, the following inferences are made. Okay, we have to see which is peripheral membrane protein and which is integral membrane protein. Which of the following option represent the combination of all correct statements? So you got two fractions upon treatment. That is, one is soluble fraction and the other is insoluble pellet. The protein X and Y are fractionated into soluble components and protein Z into insoluble pellet. So peripheral membrane proteins, they tend to collect in the water soluble components. So X and Y must be peripheral membrane proteins. If you see statement A, then protein X and Y are peripheral membrane proteins. That is correct. And protein Z, it is found in the insoluble pellet component. So naturally it is an integral membrane protein. So the answer here is option 1, that is A and B. Then next one. The melting curves shown below are for two double-stranded DNA molecules of the same length. Fraction single-stranded DNA temperature at T degrees Celsius. So they have given the Tm here, and Tm is the temperature at which one half of the DNA duplex will dissociate to become single-stranded. So for DNA 1, the Tm is over here, somewhere here. This is the temperature at which half of it will be single-stranded. So it will be somewhere here, and for DNA 2 it is more. So DNA 1 is having low melting temperature and has low GC content and high AT content, DNA 1. Whereas DNA 2 has high melting temperature. So Tm is more for DNA 2 and it has high GC content. So which of the following statements about these DNA molecules is correct? DNA 1 has lower AT content than DNA 2. That is not correct, because DNA 1 is having higher AT content than DNA 2. DNA 1 has lower GC content than DNA 2. That is correct. That is why DNA 2 is having higher Tm than DNA 1. The DNA 1 solution has a sequence independent double-stranded DNA binding protein. That is not possible with this experiment. DNA 2 has high number of mismatched nucleotides. If the nucleotides were mismatched, then Tm would have been high. Would be low and not high. If there were mismatched nucleotides, then Tm would have been low, not high. So the answer here is option 2. Then next one. Following statements are made about the supercoiling of DNA. DNA supercoiling acts as a regulator of gene expression but not of genome organization. It acts as during genome organization also. So first statement is not correct. You are asked to find a combination of all correct statements. So first is not correct. Circular DNAs found in mitochondria, viruses and bacteria are invariably negatively supercoiled. That is correct. A moving RNA polymerase generate positive superhelical tension in the DNA in front of it and negative helical tension behind it. That's also correct. Human topoisomerase 1 cut both the strands. No, human topoisomerase 1 cut only one strand of the supercoiled DNA. So D is wrong. Human topoisomerase 2 cuts make transient break in single strands of the DNA, not single strands, both the strands. So E is also not correct. So the correct answer here is B and C. That gives you option 4 as your answer. B and C only. Then next one. Microtubule cytoskeleton utilizes some accessory proteins to regulate microtubule dynamics. Accessory proteins are given in column X and their typical function in column Y. So gamma TURC. That is 3. A nucleate assembly and remains associated with the minus end. A3. So that leaves out 1 and 4. Then XMAP 215, that is found in synopsis. It is stabilizes plus end and promotes rapid microtubule growth. B1. Then C catenin severs microtubule. And augmin D2 helps in microtubule branching. Yes. Helps in microtubule branching. So the answer here is option 2. That is A3, B1, C4 and D2. Then next one. During metaphase to anaphase transition, complete removal of cohesin allows sister chromatids to separate and move to opposite poles of the spindle. Given below are few proteins or protein complexes involved in mitotic progression. Which one of the following options represent the protein or protein complexes involved in cohesin removal at the onset of anaphase during mitosis? So you have to see which one will help in the removal of cohesin. Because during metaphase to anaphase transition, complete removal of cohesin allows sister chromatid to separate. So separase if you see, it cleaves the cohesins that link the sister chromatids. And it will trigger sister chromatid separation. So cohesin is there. Then there is something called as securin. It is an inhibitor of this separase. Now this inhibitory action of the separase, it is removed by CDC20 and APCC. It promotes that these two, they promote the degradation of securin. So separase can act on the cohesin. So A, B, C are involved. So answer here is option 3. Then next one. Protein transport into endoplasmic reticulum is co-translational and proteins are inserted via an aqueous channel into the endoplasmic reticulum called transcolon. This can be studied using microsomes in an in vitro translation setup. Statements given below are possible outcomes when salt conductance is measured in this system. So if the channel is open, salt conductance will take place and the channel is open only during co-translation. So microsomes do not show any conductance of salt ions when isolated from the cells. Yes, that is correct. Because microsomes are isolated. There is no co-translation taking place. The channel will remain closed and salt conductance also will not take place. So A is correct. Addition of puromycin will lead to increased salt conductance. Puromycin causes premature chain termination and result in truncated protein. The channel it remains open there, so increased salt conductance will be observed upon addition of puromycin. Addition of puromycin will have no effect on salt conductance. That is also not correct. Which one of the following option has the combination of all correct statements? So A and B is the answer. Answer is option 2. Then next is regarding Fragile X syndrome. So Fragile X syndrome, it is a genetic disorder which occurs as a result of mutation of the FMR1 gene. The FMR1 gene, it codes for a protein called as the FMRP, and this protein, it plays a role in the development of synapses. So we will see accordingly, I will explain. So which of the following provides all correct statements? That is what they have asked. It is caused due to increased number of CGG trinucleotides greater than 200 repeats in the 5' UTR of FMR1 gene. That is correct. That is why that is the cause of Fragile X syndrome. Unaffected people do not show any CGG repeat. No. Unaffected people, the FMR1 gene contains 5 to 44 repeats of the sequence CGG. So B is not correct. Expanded numbers of CGG repeat in the 5' UTR of FMR1 transcript causes its premature degradation. That is also not correct. There is methylation of the CGG repeat expansion and FMR1 promoter, leading to silencing of the FMR1 gene and it results in lack of product formation. Then for D, expansion over 200 repeats lead to methylation of the FMR1 promoter. That is correct. Fragile appearance of X chromosome developed due to dissociation of non-histone proteins from the FMR1 locus. That is also not correct. The methylation of FMR1 in the chromosome results in constriction of the X chromosome which appear fragile under the microscope. That is why it is having fragile appearance of the X chromosome. So E is also not correct. So the correct answer here is A and D. Answer is option 4. Then next one. The following DNA molecules are provided as substrate for replication by DNA polymerase 3. Which one of the following option lists the molecule that cannot function as substrate for DNA polymerase 3? So for DNA polymerase to act, it should be single-stranded template. Then primer should be present and it should have free 3' end. So if you see here option A, it is a single-stranded template is there. Primer is there with a free 3' OH. So it will be act as a substrate for DNA polymerase 3. Now B also, if you see, it is having a free 3' OH and a template is there, a primer is there. So B also will act as a substrate for DNA polymerase 3. But if you see C, it is having 5' end only which is free. So it will not act as a template. Here it is nick is produced. Nick is translated by DNA polymerase 1, not DNA polymerase 3. So here also it will not act. So C and D are the ones which cannot act as substrate for DNA polymerase 3. Answer is option 3.